Welcome to Exploring Science. We're just going to be logging everyone in here for a few minutes. So while you wait, we have a little activity. This is actually our activity that we asked students to think about last week. So there was recently another space launch. So a couple of astronauts went up into space last week. So we thought it would be cool to think about how everyday actions might be different in space. So there's a lot of different things in space. There's no gravity like there is here on Earth. So how might that affect some of your everyday activities? Really simple things like how would you brush your teeth or shower? Normally water falls on you from the shower head. So what happens in space where you don't have the gravity pulling that water down? So I put together a few videos here that we can watch. And if you found anything while you were searching that you thought was really interesting, maybe you can put that in the chat too, and we can all hear about what you found, or maybe some cool astronauts that you follow. So our first one is about exercising in space. And this is a video, which is actually a really cool video. It's a long video, so we won't watch all of it. But the commander of the space station in 2012, Sunita Williams, gave us a whole tour of the entire space station, which was really interesting. And for part of it, she showed us how she exercises in space.
Another piece of exercise device. This is what we call the A-Red. And with this, you can lift weights based on vacuum in these cylinders, just like you do at home at a gym. For example, if I wanted to do a squat, I could do it like this. Oops. I should need some weight on this thing. I'm not lifting with anything, so one of the cool things you could do things that you can't do at home. For example, single leg squats with just one leg, because the other leg, you won't fall over. So all sorts of stuff. You can also do bench press. We have a bench that we could add on here, so you can work on your beach muscles.
Yeah, awesome. So something I hadn't really thought about is how weights are so much different in space. You can't just be lifting weights, because actually a lot of the workout you get from lifting weights comes from gravity. So when you're on the space station and you don't have gravity, you have to think of new ways that you can work out. And so if you want to check out this whole video, you can go to this link here. Maybe someone could put that in the chat as well. That would be awesome. And so this is one of the prompts we actually asked last week, was how would you shower in space? So another astronaut, Karen Nyberg, demonstrated how she washes her hair in space. Try and make sure it's not too
Start by just putting some hot water, squirting it onto my scalp. And I have a mirror here so I can kind of watch what I'm doing. Sometimes the water gets away from you and you try and catch as much as you can. Then I just work the water up through to the ends of my hair. And I take my no-rinse shampoo and squirt it also on the scalp, just a little bit, and rub it in. Again, kind of working it out to the ends. And sometimes I'll actually take my comb to help work it all the way to the ends. And I like to take my towel while I have the shampoo in there and just kind of work it. Because without standing under running water, you kind of need to use the towel a little bit to help get some of the dirt out. I like to follow that by a little more water. It's called no-rinse shampoo, but I think it's best if you use a little water with it.
Yeah, so as you can see, without the shower able to rinse out all of your shampoo, and the shower doesn't or the water doesn't actually fall, you have to use the surface tension to get the water to stick to things and get it to absorb into your hair. So it's a lot different. And so I have one more video here. And this is actually not specifically in space, but some advice specifically for middle schoolers. So I thought that that was really cool, because he's talking directly to all of you who are joining us today. And this is an astronaut. He's actually currently still with NASA. So he might be going up into space sometime soon on one of the next few missions. And his name is Victor Glover. And so we can listen to his whole interview here if you want, but specifically we're going to listen to what he says for middle schoolers.
Be a lifelong learner and be a good person. Look for challenges. Go out and seek those hard things. Learn a foreign language, learn to computer program, learn to go out and do hard things. For example, if giving speeches makes you nervous, you ought to be standing up here right next to me. Start off just like in the gym. You don't go to the gym and put 300 pounds on the bar and try to lift it. You put 10, then 20, and you work your way up. Stand in the mirror and make a speech. Then make a speech in front of your brother and sister, and then make a speech in front of your mom and dad, and then your teacher, and then your classroom, and before you know it, you'll be making an address on the mall like the president at the inauguration, and you won't be nervous, or at least that nervous energy, you'll be able to funnel it. Extremely important. You know, one of the things you learn working around a bunch of engineers is how important it is to be able to communicate. You could have the greatest design. This is going to solve these amazing problems, but if you can't communicate that, you may be the designer, but the folks who have to build it need to understand what's in your head, and that's what communication is. So it's extremely important to be able to communicate. And just like you practice football, just like you draft and you practice math and you practice computer programming, you have to do it in order to do it well. Communication, written and verbal, are things that you have to practice as well. It's interesting, you show up in this flight suit, you know, with that title, that job title, and people say, hell, you're smart, and they think about success. But I can tell you, most folks that have that title and wear this uniform have probably failed at more things. I have failed at more things than your average person. And so you're going to find something in this line of work, or anything in life, anything you really try to step out there and do, you need to step out on faith, because at some point you're going to find challenges, but they're not there for us to turn away from. Those are where we get better. You go to the gym, you put weight on the bar. You put resistance, because that's what makes you stronger.
Awesome. So as I mentioned before, Victor Glover is a current astronaut, and he's actually on Twitter, if anyone ever goes on Twitter. And he posts about his journey to being an astronaut, what it's like to be an astronaut with NASA, how he got there, and kind of the things that he's doing, as well as just what is going on with his life. Because as we mentioned a little bit last week, science is a part of your life, and all of your life really is involved in how you become a scientist and how you operate in science. So he has some really cool stories about that. And there's a lot of astronauts actually nowadays on Twitter and YouTube. So if you look them up, you can hear all sorts of different stories of how different people found their way to be astronauts, if that's something that you're interested in. So welcome everyone to Exploring Science. It's great to have you all here. This is a collaborative event between two groups that work at Yale, a group called Open Labs, which normally puts on something called the Science Cafe, if you've ever heard of that. And in normal times where we can all see each other, we like to have those events and bring our speakers together and do some more hands-on interactive things. And then the Flip Science Fair, which is what we're going to be starting with today, is actually someone presenting their poster to all of you. So in a lot of science fairs that you may have been a part of, you present to other scientists, and in the Flip Science Fair, we're going to be presenting to you. So our speakers today, our first speaker is Mike, and he's a graduate student in pharmacology, and he's going to tell us about treating cancer by making cells throw out cancer proteins. And then our second talk will be by Rebecca. She's a graduate student in neuroscience, and she is going to talk about memories and how the smell system affects memories in flies. So thank you, and we hope you enjoy our event.
Great. Well, hello, everyone. Thank you for tuning in. So as it's already been said, my name is Mike. I'm a graduate student here at Yale. Just very quickly, some background. So I'm a Connecticut native. I grew up in Windsor, Connecticut, just north of Hartford. And I also went to the University of Connecticut for my undergrad before I came here to Yale. And so today I want to tell you about something that I've been working on for the last couple of years, which is being able to throw away cancer cells using the garbage disposal that all of our cells have. And so I study pharmacology here at Yale, which is the study of how drugs affect your body. And this is a brand new type of drug that's being used to treat cancer right now. And so when you have something that gets old and you want to throw it away, you throw it in a trash can, right? And so cells do the same thing. So inside all of your cells, there are proteins, and proteins are little machines that allow cells to carry out their function, their jobs. And so when these proteins get old, or if they don't work properly, the cell needs a way to throw them away. And so there's this whole garbage disposal system that's inside the cell. So there's three main parts. We have this over here, which is the trash sorter, and the science term for this is an E3 ligase. And so this is a protein that tells the cell what proteins should be thrown away. Then we have this other protein over here, which acts as a trash label, and it's called ubiquitin by scientists. And so this protein actually labels the trash. So the trash sorter takes this label and puts it on the protein. And once proteins to be thrown away have this label on it, the trash can within the cell, called the proteasome, can then throw away the proteins. And this is done by chopping them up into little bits so that they can't work anymore. And so scientists have found a way to take over this garbage disposal system to throw away any protein that they want. And they do this using this right here, which is a Protec. And so a Protec is a drug that acts as a trash magnet. And so you can see that there's two pieces to it. It has two different heads. On one head, it can bind to that trash sorter, and on the other head, it can bind to whatever protein you want to throw away. So if we look to the right of this dashed line, this is kind of what happens in a cell when it has a Protec inside it. So normally, the trash sorter is not near the protein that scientists want to throw away. And one example of this type of a protein that we would want to throw away is something called KRAS, which the name isn't important, but what it does is it helps tumor cells grow. So it helps cancers grow. And so scientists want to be able to throw it away so that we can stop tumors from growing. And so what happens is when we take our Protec and we feed it to the cell, it'll then act like a magnet and it'll attract the trash sorter and that protein we want to throw away, that KRAS protein, and it will cause the trash sorter to label the protein. Then the proteasome, or trash can find the protein and chop it up into little bits. And so the way that I'm able to see this all happening in a cell is over here. I use a technique called immunoblotting. And so if we break down the word, immuno has to do with your immune system, and so it uses antibodies. And blotting is just a way for scientists to see proteins outside of the cell. And so the materials that I use are a cell, so I need the cell to see its proteins. And you can see that there's all these little KRAS proteins inside the cell. I then use something called lysis buffer, which is essentially soap. And so what the soap can do is it can break up the fat that surrounds the cell. So all cells, including bacteria, have fats around them. And so when you wash your hands with soap, the soap breaks up the fat around the bacteria and kills it. And that's why it's good to wash your hands. And so the same thing applies to cells in the lab. We can use the soap to break open the cell, and then we can look at all the proteins inside. And so the last thing that I use is where that immuno part of immunoblotting comes in, which is an antibody detector. And so this antibody can specifically recognize the protein that we're trying to throw away, and then it has a detector on it where we can see how much of the protein is there. So on the bottom here, you can see an example of what this immunoblot might look like. And the darker the bands, the more that's being detected by our antibody and the more protein that is around. So that's what these arrows mean. On the left, there's high protein. On the right, there's low protein because we've added our Protec in. So this wedge here, this red triangle, tells us how much Protec is around. So on the left, there isn't a lot of Protec. And on the right, there is a lot of Protec. And when there's a lot of Protec around, what do people think will happen? I'll look at the chat to see if anyone can answer. So if we have a lot of Protec around, what do people think will happen? Exactly, exactly. So I just saw in the chat that KRAS will go down, which is exactly what you see here. And we know that that's happening because this band gets lighter and lighter. And so if we go to the center here, we can see some blots that I actually did in the lab. And one thing that I didn't mention over here is that we're not just looking at KRAS, we're also looking at this other protein, which over here is called the control, and over here is called tubulin. They're the same thing. And so what this is we want to make sure that the Protec is only throwing away KRAS and isn't throwing away this other control protein, in this case tubulin. And we also want to make sure that we're adding the same amount of protein to each of the lanes that we're running through the gel to make sure that it's the Protec that's causing the protein to go away, and not just that I'm adding less protein to the gel. And so you can see here that when we don't have a lot of Protec around, we see these dark black bands. And then as we move to the right, as the amount of Protec increases, we can see that band get lighter and lighter, even though our control, our tubulin, is staying the same. And so what we can conclude from this is that KRAS Protecs can cause protein loss in cancer cells, so we can throw away those proteins. Now, the next thing that I wanted to do in the lab was I wanted to make sure that this drug was working the way that we thought it was working. And so for that, I wanted to make sure that the proteasome, it was dependent on the proteasome, or this trash can. And so I'm giving away the answer here, but what would happen if the lid on the trash can is closed and you can't actually throw away KRAS? Exactly. So it would build up, which is what I just saw in the chat. So that's what you can see here. So if we go, we look at all the lanes. In this lane, we don't have any Protec, we don't have any of our drug, and we also don't have to worry about the proteasome, so the KRAS is around. Then we add our drug in, and the lid is off of the trash can, so we can chew up the protein, and KRAS goes away. But then if I close the trash can we can do this by using another drug that stops the proteasome from working and kind of, shuts the lid on it. Even if I add in the Protec, we don't see the protein go away. And so this is good because it means the drug is working the way we want it to work, and it means it's specific for what we're trying to do. And so just some conclusions that we can make from those few figures I showed you. So we can throw away KRAS, which is an important cancer protein, and this is a big deal because someday in the future, we could use a drug like this in patients. And although there are Protecs that are actually currently being used in patients, but they cause the cancer cell to throw away other proteins, not KRAS. But in the future, it could be KRAS. And we can also conclude that the proteasome, or that trash can, is necessary. So this is working in the way we designed it. And so we're able to take over the cell and do what we want. And so thank you for listening. And I'm excited to answer some of your questions.
Thank you so much for presenting. So I'm going to ask some of the questions from the chat that we've collected to you.
Great.
So I'll start with some questions about this drug. I guess these are kind of a couple related questions. How long would it take this drug to work? Is it expensive? Is it available for everybody who needs it? Or is it still like very experimental?
So this specific drug is experimental, but the other types of drugs are in what's called clinical trials. And so they're being tried in humans, but they're not officially approved yet by this thing called the FDA, which is the Food and Drug Administration, which controls when medicines get approved. So I don't know how expensive it will be, but unfortunately, like a lot of medicines for cancer, it will probably be very expensive. To quickly address like how fast it works, in the lab, when I feed this to cells, I can see KRAS go away within 2 hours. So it works pretty fast, but that doesn't mean that it'll take 2 hours for it to get rid of a tumor in someone's body. It will probably take longer.
Okay, thank you. Another question. So, um, why why has this drug sort of been recently developed? Is there a reason it hasn't been used before? Like, does it have some kind of bad side effects?
So that's a great question. So actually, the reason that this was developed is because it should have less side effects, because it's more specific than the drugs that are used now. And so the reason it didn't exist before is because people didn't think it was possible to take over the garbage disposal system. But Craig Cruz, whose name is up here, who's the professor that I work under at Yale, actually discovered that this whole technology works. And the reason it hasn't been used before is also because it took like 20 years to actually turn the idea into something that could be used in people. So it takes a long time, but there should be less side effects because it's like really specific for the protein that you're trying to throw away.
Great. And a final question: why are there so many kinds of cancers? Like, why do we have to have all these different methods to fight cancer?
That's a great question. And it would probably take me as long as we have the entire session today to talk about that. But just briefly, there are so many types of cancers because there are so many types of cells in your body, and all of those cells can become tumorigenic, which means they can start growing tumors, they can start becoming cancers. And also, you have a ton of genes in your body, which are the things that make the proteins in your DNA. And almost all of those can get mutated in such a way that can cause cancer. And there's different combinations, and those different combinations cause different types of cancer, which need different types of medicines to treat. So just the amount of cells in your body plus the amount of possible mutations means we need a lot of drugs to try to treat cancer.
Okay, thank you so much.
Our next talk is going to be very exciting. It's by a graduate student here at Yale, Rebecca, and she's going to be telling us what we can learn about learning, but from fruit flies.
So I'm Rebecca. I'm a first year PhD student in neuroscience, and I'm very excited to talk to you about what bug brains can teach you about yourself. Let's see. So picture this. When you were little, you'd like to bake these nice chocolatey cookies with your mom every Christmas. Time passed, you grew up, and you kind of forgot about that for a while. But one day, you walk past this bakery, and you smell this nice chocolatey aroma. Suddenly, you're transported back to those times you were baking these chocolatey cookies with your mom every Christmas. So what are these positive associative memories? And how are they formed? So it turns out the fruit fly, Drosophila, does the exact same thing that we do. They can learn to associate a certain smell with a positive thing like sugar. And so what I'm going to be talking to you a little bit about today is how we can understand long-term memory in fruit flies, and also how our understanding of learning in flies can shed a little bit of light on our own learning. So this is a little bit like Rick's talk, where he was talking about how learning in mice can help us understand ourselves. And here I will tell you how even flies can. So that's even simpler animals. So a little bit about me first. So for the longest time, I thought I was going to be a big mammal wildlife biologist. So for a lot of my undergrad years, when I was in college, I would be out in the field studying animals like Arctic foxes with questions like, how does the behavior of an Arctic fox change according to different predators it has? And then I started to become a little bit curious about what happens in the brain that can lead to this change in behavior that we can see in our data. And so there's an issue with that. You can't exactly put wild foxes in the lab, and you can't really do things to their brain because they're wild animals and they have they're protected in that way. But that's how I got really interested in fruit flies, because they're very common lab animals. They've been lab animals for a very, very long time for scientists. And the great thing about fruit flies is that we have a very good idea of how their biological building blocks, so how their genes and their cells contribute to how they are and how they change over time. So for example, here, the different colors correspond to genes that actually are responsible for the formation of different body parts of the fly. So we can kind of understand how each body part is formed by these genes. So let's go back to memory. What do we know about how memory works? So scientists have been trying to figure this out for years. Maybe some of you have heard that, or you haven't even had this happen to you if you've been in some experiments where they'll have you sit and the scientists will put these funny hats on you, and they'll get these waves of activity at different frequencies. And scientists have learned that some of these frequencies are associated with certain forms of memory. For example, like working memory. And that's what you use when you try to remember, for example, a phone number someone just told you, but that you may forget like 10 minutes later. But what's important to keep in mind about these different waves of activity is that they come from maybe thousands to millions of cells. So that's a lot of activity from a lot of different cells. But what I wanted to know is, how can we understand memory from the level of one cell, one brain cell, one neuron to the next? So it turns out fruit flies are very great for this. You have 86 billion brain cells. What do you guys think? How many do you guys think flies have? Okay, so we've got everything from 50 billion to 30. It turns out it's a little bit in between those two. They have around just over 100,000. So actually someone guessed 200,000. So that was pretty close. Which actually, if you think about it, for a tiny fly, that's actually a lot, but it still means that compared to 86 billion, it means that circuits, so connections of cells that do similar things between flies and humans, tend to be much simpler in flies, which is very nice because it means that studying flies can get us closer to our goal of trying to understand memory from one neuron to the next. So although flies have much simpler brains, like I kind of previewed, they can actually do similar things that we can do. They can associate one smell with a given context, and then they can change their behavior accordingly. For example, if they smell that smell again, they can go towards it if it was associated with something good, or they can go away from it if it was associated with something bad. So how does that actually work? So what we're going to do now is take a little field trip inside the brain of the fly, and specifically inside the memory center, which is called the mushroom body, because it kind of looks like a mushroom if you look at it closely. So where is smell and emotion in the mushroom body of the fly brain? Turns out it's actually quite well organized. You have smell come in, and smell information is relayed by different neurons until it goes out to influence fly motion. And along the way, you can see these colored neurons here. You see the red, green, and blue. So over time, scientists have found that if you paired something negative with a smell, so if you give a smell to a fly and at the same time you give it a foot shock, for example, a little electric shock, then these blue and red neurons would be active. And next time the fly smells that smell, they would move away from it. And similarly, they found that if they paired something really good with a smell, so if they gave the fly sugar when they gave the fly a smell, then these green PAM neurons would be active. And that next time the fly is faced with that smell that was paired with sugar, then they would go towards that smell. So this is an overview, but what exactly is changing in the smell system for the fly to be able to make these decisions next time it encounters the smell? So we can try to look at a kind of simplified version of this system. We have these Kenyan cells that relay the odor. We also have these PAM cells, and if you remember, they're in charge of bringing in that positive context if you pair something good with a smell. And then these Kenyan cells actually connect to mushroom body olfactory neurons, which connect smell information to influence fly motion. So if we want to understand what is changing in this series of neurons to allow a memory to form, what neuroscientists really like to do is they like to mess with the system. And so what do you guys think would happen? Well, knowing that first, that PAM neurons are important for putting in that positive context if you pair sugar with an odor. What do you guys think will happen if before you pair sugar with an odor, you actually stop this neuron from working so that the flies don't have PAM working at all before they're shown sugar and a smell? What do you guys think will happen once we show the fly the smell again? And they're supposed to have remembered that that smell is paired with sugar. what do you think will happen to the fly? I think most of you got it. So it turns out that you can kind of look at the data here. The blue flies are the ones that have PAM intact, and then the orange ones are the flies that did not have PAM working. Turns out that flies, even though they're hungry, if they don't have PAM working, they won't be around that smell that was associated with sugar, which basically suggests that they're not able to have that memory formed. They were not able to associate sugar with that odor. So how does that work? How can we understand this? So it turns out that actually PAM neurons communicate to Kenyan cells below here by this chemical called dopamine. And so it turns out that if you stop Kenyan cells from receiving the message from PAM neurons, then you get similar data. You get that these flies can't remember that this smell was paired with sugar. So they have no idea that they're supposed to go to it. And so it turns out that through these experiments, we can understand that PAM, and specifically the dopamine that PAM neurons give to Kenyan cells, are necessary for memories for sugar odor association to form. So we spoke a little bit about how PAM speaks to Kenyan cells to form a memory, but what happens downstream? How do we eventually get changes in how Kenyan cells speak to MBON neurons to eventually have the fly change its behavior, so move towards this sugar-associated smell? So one way to look at that is to look at the activity of MBON neurons before and after you pair the sugar with the odor. And so now I'm going to tell you guys a little bit about this cool trick that neuroscientists use. And it's based on the idea that neurons use calcium to send messages to one another. That means that if you have a lot of calcium in your neuron, it's going to be sending a lot of messages to its neighbor. And if you don't have a lot, then your neuron is not very active and it won't be sending a lot of messages. So it turns out that what neuroscientists like to do is they like to put in this protein in the neuron that they want to study. And Grace told you a little bit about this protein already, that it can glow green, which is very useful. You can actually visualize it. And in this case, this protein will glow green if it's in contact with calcium. So basically, if we look at this graph here, before this neuron gets any kind of stimulus, any kind of message, it doesn't glow at all. It's not very active. There's no calcium. As soon as it gets a message from its neighbor, then it starts to glow green. And the bigger this glow, the bigger this fluorescence change, the bigger we can say that the neuron's response is to a given stimulus. So that's very useful because now we have visuals to understand how the activity of MBON neurons can change before and after a sugar odor association. And so what we can see from results is that when you pair, before you pair any odors with sugar, when you show the fly this blue odor here and then this red odor, before any pairings have happened, then this MBON neuron, as seen through this activity here, it reacts similarly to both odors, which is expected because it's in the odor pathway and it should react to odors. But if you pair sugar with this blue odor, then you actually get a greater response in this MBON neuron. But this greater response compared to the odor that was not paired with sugar at all, which is this red odor. And so this suggests that one consequence of the sugar odor association we already looked at here, but now we're looking at this area in the MBON neurons, is that you have greater activity there, which basically suggests that we have a greater connection strength between Kenyan cells and MBON neurons after this sugar odor association has been done. But so now we have a final step missing. How does this impact fly behavior? Great, we have more active MBON neurons in response to sugar odor associated smells, but how does the fly behave differently? So you can kind of think of dopamine from PAM neurons as a dial of the connection strength between Kenyan cells and MBON neurons. So after you pair this odor with sugar, you get this greater connection strength between Kenyan cells and MBON neurons. And then the reason why that's important is that then you can increase approach behavior towards that smell. And so we can say that at least a component of the memory, the sugar odor memory, is in this increased connection strength between Kenyan cells and MBON neurons. And so how does this relate to us? You know, we talked about how we have so many more neurons than flies. So why would this finding in this increased connection strength relate to how our memory works with smell? So just like the overall brain, there's probably many more neurons in the smell system in humans and in the memory system in humans. But remember from the beginning of the talk, our brains are made of the same building blocks. Whether you're a fly, whether you're a human or you're a rhinoceros, your brain is made of neurons. And so that means that just like the fly that is associating odor with sugar, changes in the connection strength between neurons probably also underlie why you can associate smells with different positive memories that you had in your childhood, like baking cookies with your mom when you were little. And so I hope I told you a little bit about how we can use these very simple tiny animals to yield greater information about ourselves and about our past. And if you're anything like me, maybe before you thought of flies of something like this, and maybe you're thinking of flies as something that's maybe too close to us for comfort. So, very similar to what we're doing every day. And with that, thank you so much. And I'd love to answer any questions that you guys may have. And thanks for listening.
That was a great presentation, Rebecca. I have a couple questions from the chat for you.
Sure.
The first question is, how can flies remember these smells when they smell so many things?
Yeah, so that's a really good point. Flies are probably just like you're probably bombarded with huge amounts of smells every second, not even throughout the day. So it's probably a good idea for flies to be around smells that will predict things that will help them survive. So in an environment that is constantly changing, where they can't necessarily know where their next food source is going to come from, it's probably a good idea for the fly to have some really solid way of being able to remember that a certain odor was paired with something that it needs, like food, for example. So we can probably bet that if you present something with something like a sugar or some other food they really like with a smell, they're probably going to be picking that out from a lot of different smells because they want to be eating as much food as they can. So that's definitely a good way to do it. You can also pair with things that they really want to get away from, because again, that's also helpful for survival. So if you pair something with a smell electric shock, the fly will not want to be around that smell. Even though that smell could be relatively similar to other smells, it'll sure as hell remember that one because it doesn't want to die, or it needs to survive.
Great. And another question, you talked about calcium being necessary. Where does the calcium come from? Is it by digestion? How does the calcium get inside the flies?
Yeah, so these neurons are sitting in this fluid essentially, and the way that they work is that they have these little trapdoors that open and that let in certain chemicals at certain times. And a lot of these chemicals have different roles, but one of them is to help the neuron release chemicals to its neighbor, and that's exactly what calcium does. So when the calcium, essentially that calcium trapdoor in the neuron opens, that's like a go signal for the cell. As soon as calcium is in there, you're going to have a bunch of little chemicals packed in membranes that is going to go to the neuron that is connected to your first neuron. And so that's the specific role of this chemical. But it's important that the trapdoor for calcium is regulated, because you can imagine you don't want your neuron to be sending random messages all the time. So it's only when that neuron is going to send a message that you want calcium to be there. It's very powerful.
Great. And one last question. So you're talking a lot about neuron to neuron communication. If there are multiple neurons involved in this communication, will the number of neurons affect the strength of the connection and the reaction to the smell?
Oh, that's interesting. The number of neurons impact the strength of the connection. So that's interesting. Right now, what we were looking in isolation, 2 neurons out of a much bigger network, right? So that's a really good question. There's probably a lot more complexity to this network than what we can understand, but that's of course why we use such a simple animal like the fly. But you can imagine that it's probably not just that, right? There's probably one component of the memory that we've isolated, but there's probably this other branch of, for example, another neuron that could be impacting the neuron that I'm studying that could also be contributing to an effect that we see. So all that we have is we've isolated something that impacts memory, and also some trace of the memory, but that's by all means not the only trace of that memory. There's probably some other some other elaborations that come from other connections of other neurons. This does not exist in isolation, this little circuit that we're looking at.
Hey everybody, so we're back here with Rebecca taking a few more questions after exploring science about fly brains that I'm going to ask her. So we're going to start kind of focused on the fly and then we're going to spread out. So my first question is, Rebecca, can flies forget things?
Yeah, totally. Just the way that you can forget things, you can imagine that even tiny little flies can forget things. So when we talked about this odor that was paired with sugar, if you give that same odor and you don't pair it with sugar anymore, and you keep showing it to the fly, and the fly keeps thinking there'll be sugar paired with it, and then there isn't, well, over time, that association is going to get weaker and weaker, so that after several times of this, if you just show the fly this odor, then it will no longer have that association. So that is something that flies can experience too. So that's also a way of forgetting the association, just like you forget memories as well.
Wonderful. And our second question is, how do you actually study these flies? I mean, flies are pretty small, right? Like, how would you actually look at them in the lab?
Yeah. So they're tiny. They're much smaller than your fingernails. They're like a tiny fraction of your fingernail. That's how big they are. So what way that we study them is that we have these big microscopes that allow us to and then you have the big microscopes. Then you can see the image of the fly brain blown up on your computer, and then you can use tools like this protein that lights up. That lights up green in contact with calcium. So that can help us not only just look at the cell, but look at its activity. So those are the different tools that we have, but there's so many other tools that scientists have developed over time. The really cool ones, for example, you can control the activity of neurons too. Optogenetics, like Rick must have told you about as well, that you can also use in flies. So there are a lot of different tools available, but they also involve visualization with microscopes. That's definitely a big one when your animal is so tiny.
Wonderful. And our final question, kind of a broad question, Rebecca. You've told us a lot about flies. What about other insects, bees and wasps? Do they also work in the same way?
Yeah, so bees and wasps are not as commonly used as flies. So flies have been used by many years by many different kinds of scientists, so geneticists, besides neuroscientists. And so that means that there's a lot of tools that we can use to study flies, but that doesn't mean that other animals don't have the brain structures that flies have. They probably actually do, because if you think about it, the fact that you can associate things, the fact that mice can associate things means that the tools that you have in your brain to form memories are probably really widespread throughout different organisms. So it's probably a good guess that the way that a wasp would associate sugar with a smell is probably not so different from the way that a fly would associate sugar with a smell. So probably similar, but it's just we haven't tested a lot of these things. There's a lot left to look at.
All right, wonderful. Well, thanks again, Rebecca, for talking to us and exploring science. And everybody watching Theoretically, I guess we hope to see you next week for more talks.
Thanks, guys.
All right, so we're back catching up with Mike after his great presentation today. He talked about the trash can of the cell and how his lab is able to use it to fight off cancer. So you guys had a lot of really great questions in the chat that we weren't really able to get to. So kind of separated into several subgroups of questions. So we'll start with the first one, which is a bit about more background on cancer. So Mike, what is cancer? Where does it come from? And why is it so bad? What makes it so hard to treat?
Well, so the reason why cancer is so hard to treat is because it actually comes from your cells. So a cancer is just when one of your cells starts growing out of control, and your immune system can't stop it from growing out of control. And so this happens from things like mutations that I mentioned during the presentation. So when there's changes to the DNA, the cell can kind of go crazy and can grow uncontrolled and then forms a mass of cells. And when that happens, that can cause your organs not to work properly. And if enough of your organs don't work properly, there's not a good outcome for you.
Okay, cool. And as far as what types of treatments currently exist, so, what are they? How does this drug that your lab is developing compare to some of these?
So there's a lot of different treatments that are out there. I'll give a couple broad category. So there's radiation. So you can use things like high-powered X-rays to kind of zap the cells and try to stop them from growing. There are chemotherapies, which a lot of people are familiar with, although underneath chemotherapies, there's a lot of different kinds of drugs. So generally speaking, there are drugs that stop the machinery that replicates DNA from working. There's drugs that stop the cytoskeleton, which I mentioned during the presentation, from working, so the cell can't move, it can't divide anymore if it can't move. And there are also drugs now that are antibody-based, and the idea there is to try to help your immune system find the cancer and then have your body destroy it. So the drug that I presented, these Protec molecules differ because they're kind of based on a different idea of medicine. So most chemotherapies have to stick to whatever they're binding to, whatever they're stopping from working for a really long time. Whereas the molecules that I talked to you about today, they only have to stick to the proteins long enough for that trash label to get put on them. And so because of that, they can actually work in what's called a catalytic mechanism. And so what that means is that you can actually get rid of more of the protein with one molecule of a Protec than you can with one molecule of a chemotherapy. And so what we're hoping is that because of that, we can give less of the drug to patients, and then there'll be less side effects and it'll work better.
Awesome. So kind of along the line of your drug specifically, is it available for patients yet? How expensive is it? Can it help all patients? And, why might it help or why might it not help all patients?
So my drug that I showed today in the presentation is not available for humans. It's still being tested in the lab, but molecules like my drug that cause cancer cells to throw away other proteins, they're called the androgen receptor and the estrogen receptor, and those are important for hormone signaling and things like that, and are important in things like prostate and ovarian cancer. Protacs for those proteins are currently being tested in clinical trials. So people can't go out and buy those quite yet, but they're being tested so that people eventually can do that. And real people with cancer are taking those Protacs right now. There's a company called Arvinas, which my boss Craig Cruz started. And so if you're interested, you can read up more on them. And so this Protec might not be able to help all patients, and that's because it's specific for a specific version of KRAS. So it's called a KRAS G12C. And so the G12C stands for the building blocks of the machine that are changed. And so only patients who have cancers with that G12C mutation would benefit from this Protec. But the good thing about that is that we won't actually be throwing away the KRAS protein in other cells in the body where it's needed for cells to properly divide.
Awesome. And that kind of gets at the follow-up question to that, which is like, how specific is it? Does it affect non-cancerous cells? And then the possible side effects that you might encounter.
Yeah, so it could affect non-cancerous cells because it can still bind the trash sorter in other cells throughout the body. And another issue, and this is an issue with all chemotherapies, is that when you take a drug, your body does stuff to the drug. So that's called metabolism. So your body doesn't actually want the drug to be around and it tries to get rid of it. And when it makes changes to drugs, those are called metabolites. And some of these metabolites can actually be dangerous. And so that's why we have the clinical trials to try to make sure that the metabolites are not dangerous. And usually that affects your liver. But based on the clinical trials from those other Protacs I was talking about, we don't think that there'll be very harsh side effects. It'll kind of be the normal ones that you usually see with chemotherapies like nausea.
Okay, and one question that I didn't quite get an answer to previously, and you might not even know at this point, but how expensive is your drug or drugs that are like it?
So the cost, I don't really know quite yet, but based on kind of some other small molecules, which is the class of this type of drug that are out there, it could be on the order of $100,000 to $200,000 a year for treatment. So it is quite expensive, but that's because there's a lot of research and development that goes into making these. And just to give some perspective, those clinical trials I'm talking about can cost millions of dollars just for one, and you have to do at least 2 or 3 to actually get the drug approved.
And then the last set of questions that came from the chat are, how does it kill does it kill the cells, or does it slow it from spreading? And kind of tagged along with that, how fast does it work? Is this something that, can it do its own thing on its own, or is it the type of thing where you might have to combine it with other types of treatments?
So ideally, Protec should be able to work by themselves, and they should work relatively fast. I would guess giving it to a person, probably within a few months, you would see a change. When I feed it to cells in a dish, it only takes a couple of hours for the KRAS protein to be thrown away. But that doesn't translate exactly to a person. There are other factors. So it does kill the cells, and therefore the molecule is called cytotoxic. But it may not necessarily stop it from spreading. So it will kill cancer cells that are susceptible to it, so cancer cells that can be killed by it. But if there are some cells that aren't affected by it, it won't stop those from spreading, unfortunately.
Okay. So I think those are all of the big questions that I had and that came out of the chat. But are there any other points that you want to make, or things that you want the audience to take away?
I just, I really want to thank all of you for tuning in and listening to the presentation, and just to stay curious and keep asking questions.
Awesome. Thank you. And thank you everyone for listening.